Okay, well, thank you so much, Samir, for the kind introduction. Also, thank you all for participating today. I know it's quite early, but I hope that this presentation would be as exciting as our next speaker too. Okay, so I'd like to thank Covaris again for once again inviting me for this breakfast workshop. So basically today I wanted to discuss the use of AFA technology that I have actually used in order to increase proteomic coverage. And this is based on the continuation of the high throughput cell-based assay system that I have developed over the past 2.5 years. And I've presented in the past breakfast workshops, and this is a complementation on those talks. Okay, so I'll be talking about the study rationale and some background context of the work, and follow this up with our findings from the previous work that I presented, and then basically discuss the application of sonicating samples during digestion, or AFA-assisted tryptic digestion, and then highlight some of the future studies and next directions of the project, and towards the end I'll just summarize the points from the presentation. So I'm now going to give you a very brief synopsis on the work that we have been doing in our group at Cedars-Sinai, and we are based in the Heart Institute in Jenny Van Eyck's lab, and so we really use our technology to advance proteomic approaches to tackle the current challenges in precision medicine, and this primarily focuses on disease prevention, but can also help in treatment and therapy approaches. So to really establish a path to clinical proteomics, we really need to have high throughput assay systems in order to then quantify these and really have a high throughput system where we can routinely assess different drugs and investigate cellular perturbations. So we previously developed this automated high-throughput protein extraction workflow that really rapidly processed cardiomyocytes in a bottom-up proteomic approaches. And so what we did was we developed multiple workflows depending upon the peptide load acquisition gradient for different purposes. And in this particular presentation, I'll be talking about how we went one step further and added an extra step, and that is sonicating samples whilst being digested in order to increase the proteomic coverage. So some of the previous work that we have done involves looking at the cardiac ischemic reperfusion model. So in this presentation, in the previous presentation, what we did was we cultured the cardiomyocytes in order to investigate hypoxia in a detergent-free system to really test cardiac perturbation. And so in summary, pre-cultures of AC16 cells were seeded for 3 days, and as you can see, 25,000 cells were basically cultured in a 96-well plate, and then the cells were perturbed and harvested onto our Beckman I7 biomix system. And then the samples were then reduced, alkylated, and digested, and then finally the peptides were dried and acquired on the LCMS system. So we'll be focusing mostly on the digestion phase for this talk. Okay, so to summarize our previous findings, so you can see on the left, you have the volcano plot that illustrates the vast number of differentially expressed proteins lost in hypoxic cells with cobalt chloride compared to the healthy control cells. And so despite a lot of the proteins being lost, there were some upregulated proteins that we detected in our system, and those likely played supportive roles following oxygen depletion. So we then looked at the top 10 differentially expressed proteins, and as you can see, 7 of the downregulated IDs actually corresponded to ischemic indicators as reported in previous publications. And so the manuscript that I'm currently working on is almost ready for submission and really discusses all of these proteins. So we then matched these proteins on our in-house protein network interaction to map the biological processes. And so we observed a multitude of impacted molecular functions involved in DNA replication, mRNA modifications, mitochondrial organization, and particular cellular responses to oxidative stress, which was really exciting for us to observe these proteins. And then we further explored some of these metabolic pathways, and basically the top differentially expressed proteins, as expected, were downregulated. But then the 4 upregulated proteins corresponded to high-density lipoproteins that likely was a result of a coping mechanisms cardiomyocytes utilized to adapt to their oxygen-depleted state, and therefore reducing the accumulation of cholesterol and fatty acid buildup, and why many of these upregulated proteins participated in lipid metabolism. Okay, so after validating the stability of the high-throughput cell-based assay system, we actually employed several approaches to investigate if we can actually increase the proteomic coverage, because we really wanted to identify some of the lower abundant proteins as those were very significant in terms of being membrane bound, and we really wanted to understand how we could quantify them. So the most promising approach that we came up with was the incorporation of an AFA-assisted tryptic digestion, which I will discuss for the remainder of the talk. Okay, so to investigate our hypothesis on the increase in proteomic coverage from the addition of sonicating during digestion, so we tested our AC16 cells with and without sonication at 2 hours of digestion. And by that, I mean 1 hour of it is active sonication, and the other hour is the break between the different cycles. So the way it worked was the sonication system basically was basically sonicating, lysing the cells or digesting it, and then switching to the next condition during the 10-second delay, and then going back to the previous condition. So it's a switch on and off between the different conditions, because with Covaris, you have one column being sonicated and then it moves on to the next one. So this really allowed us to basically process many samples in a 96-well format, which was really the whole point of a high throughput system. Okay, so in this slide, you can see the correlogram and the density distribution showing the AFA-assisted digested samples, which really demonstrated a very tight association and a more positive correlation compared to the classically digested samples that displayed a far greater spread in their unimodal distribution. So we then further analyzed the two conditions by directly comparing the top 3 reliable replicates, and in the 3D PCA plot, the biological replicates of each condition was clustered independently, resulting in the replicates associating closely with each other. And then in the middle part of the slide, you can see the dendrogram confirming the clustering of the biological replicates. And the PCA biplot was quite interesting because it showed the replicates from the AFA-assisted digested samples are pretty much more correlated, whereby the greatest shift in the PCA dimension was actually directed by a protein predominantly detected by the AFA-assisted digested samples. Also, most of the proteins were also supervised from the biological replicates of this particular condition. Okay, so in this slide we have so I constructed the protein order of the two conditions ranked against their mass spec intensities, and so what I did was each curve represents the number of highly abundant versus lowly abundant proteins, and I did this by basically constructing a polynomial best fit to match the multilateral distribution of the data set. And the proteins that have been plotted are based on 75% being present in all of the conditions. So I denoted them as being reliable. So you can see the sonicated samples in green and the classically digested samples in blue. And as you can see, lower abundant proteins appear to be better detected with a higher intensity in terms of the signal when the corresponding samples were sonicated during digestion. And this really provided the first evidence of AFA-assisted tryptic digestion for increasing the detection of the lower abundant proteins. So to further elaborate this, at the peptide level, we plotted the intensities once again on another dynamic range graph. And this time we actually observed 18,500 peptides being detected in both of the conditions over the corresponding biological replicates. And this really verified the similar trend that we saw in the dynamic range of the proteins. So So as I mentioned before, the dotted line intersecting the polynomial trend line in each condition demonstrated the ability of detecting the specific proteins that were either high or lowly abundant proteins, and you can see that it's much higher with the AFA-assisted samples, as you can see over here. Okay, so we then went about looking at the differentially expressed proteins between the two conditions, and 47% of the total detected proteome was significant, which showed the efficiency of the developed workflow with AFA-assisted tryptic digestion. Now, on the volcano plot, there was a clear difference in sonicating samples was being digested when you compare it to the unsonicated control after 2 hours. And as you can see, there were 3 times more proteins being upregulated when the samples was indeed sonicated following a 2-hour sonication. And this was really exciting for us. So we continued our differential expression analysis, and we generated the violin plots of the top differential expressed proteins and a heat map to really compare sort of the spread. And when you look at the violin plots, only 2 of the top 10 differential expressed proteins displayed downregulation, and the remaining upregulated proteins showed a very tight spread of data. And a similar observation was seen in the heat map, where most of the upregulated proteins displayed minimal variability amongst their biological replicates. So we then assessed our approach by looking at the gene ontology annotations, and we observed 100 significant impacted proteins being involved in cell substrate junction and focal adhesion. Now, these proteins were annotated as important cell surface markers that could be targeted for drug delivery. And we also saw that a further 90 impacted proteins were actually involved in cadherin binding, and these type of transmembrane proteins allowed cells to adhere to each other to act also as potential biomarkers. Right, so after establishing the AFA-assisted tryptic digestion, we went about investigating the upper limit of detection by evaluating the peptide load. So how much more can we actually get increase the proteome coverage? So we basically loaded the peptides by increasing it by 3-fold to see the effect. And as you can see, increasing the peptide load by 3-fold significantly enhanced the detection of the proteins by about 40%. So therefore, 3 times more protein load, up to 390 nanograms, actually significantly increased the proteome load. So that was very significant in terms of what we wanted to see. Now, further differential expression analysis revealed a similar observation with both the heat map in the slide and the violin plot, displaying a 2-fold increase in the number of proteins detected in the samples loaded from, excuse me, a 390 nanogram injection. And by the way, this was over only a 45-minute acquisition gradient. So I would assume that if you were to perform it at 60 minutes or even 120 minutes, you would get significantly more proteins. And we actually performed this with the Bruker's system, and we got over 5,000 protein IDs. So this is just on the Thermo Explorers 480 with a substantially lower 45 acquisition gradient. So another important parameter to consider when assessing the digestion efficiency in any type of workflow, and in our case, the AFA-assisted tryptic approach, was the number of miscleavages. So on the left, you can see that 60% of the peptides displayed no miscleavages involving lysine or arginine, and a combined total ratio of 90% of the peptides showing no or 1 missed cleavages. So that was very significant. And when mapping the differential expressed proteins, we actually noticed that the nucleus compartment really comprised the most affected cellular region, whereby approximately 42% of the proteins identified as nuclear proteins, and this denoted a 10% increase with AFA-assisted tryptic digested treatment. So another thing that we were interested was mitochondrial proteins, because one of our sub-projects is mitochondrial biogenesis. Now, you can actually see that when we compared the different loads, you would see that there was a 2.3-fold increase when the injection volume was increased by the 3-fold injection. So here we have the ontology network of the significantly expressed differential proteins, and the highlighted proteins are in the protein interaction network extraction diagram, and you can actually see most of the identified upregulated proteins between the AFA assisted and classically digested samples involved mitochondrial-based processes, which was amazing. And when further analyzing this, most of the upregulated proteins actually played pivotal roles in mitochondrial organization and transport, and there were more upregulated proteins with greater fold change values based on mass spec intensities compared to the downregulated ones. So you can see 6 times more proteins being upregulated in the left diagram, and more than 40 upregulated proteins corresponding to mitochondrial organization and transport. Okay, so that concludes the first part of the sonication digestion slides. Now, we further, we continue our efforts in seeing whether we can actually further increase the proteome load. So what we did was we tested various AFA conditions by sort of like altering the peak intensity power and duty factor, as well as the assay incubation time, to see if we can actually increase it even more. Now, on the left, you can see all the different conditions that we tested. And on the right, you can see the bar charts representing the unique protein IDs and coefficient of variation for the different biological replicates of each data set. Now, I can't exactly mention the specific conditions because I'm also writing a paper on that would be published in a few months' time. But as demonstrated, one of the conditions displayed an additional 300 proteins whilst having a similar CV range to the other conditions, and that was of paramount interest to us. So we went to really look further onto that one particular condition. Okay, so on the Venn diagram, approximately 3/4 of the protein was shared between the replicates across the 5 tested conditions. And that indicated a 25% of the unique IDs detected based on condition, and moreover, only approximately 2% of the proteins were detected as unique, and these were exclusive to each data set that proved to show the high reproducibility as well as similarity between the tested conditions. And the PSA plot to the right also shows the samples being clustered closely together, which is also another proof of reproducibility. So the next couple of slides is all about the representative condition, and by that I mean the one condition that had 300 proteins. So we compared the top 2 AFA conditions and did a differential expression analysis. So the volcano plots basically are plotted here, and it really displays the number of proteins lost when you deviate from the optimized representative condition. And it also shows that the representative condition increased the protein coverage by approximately 15%. And these proteins were then further evaluated in the next slide. Another gene ontology annotation. And you can see a clear indication of membrane-associated proteins being detected now from our representative condition when you do the AFA-assisted tryptic digestion. And the 2 most dominant cluster of proteins from both conditions involve focal adhesion and cell substrate junction like before. And this was really significant once again. So the last slide for evaluating these, it's a heat map and violin plots. And you can see that the top 10 differentially expressed proteins basically show some interesting insights. So the UQC1 gene encoding protein, for example, encodes a mitochondrial protein required for respiratory chain and for the assembly of the ubiquinol cytochrome C reductase. And the other 2 identified upregulated proteins involved a phosphoprotein and a G-coupled protein receptor, which are really critical proteins for different higher ordered processes. So for the short-term and long-term goals, for the immediate objective, what we want to do is to integrate label-free and stable isotope labeling workflows. And one of my side projects is using AHA labeling to characterize newly synthesized proteins. And I really want to integrate that onto the high-throughput platform. And that would then allow me to study multiple protein folding and post-translational modifications. And this is exactly why it's important to really detect more phosphoproteins and other proteins with post-translational modification. Now, the long-term vision is, of course, to develop targeted assays on the top peptide candidates from whatever biological perturbation that you do through MRM or SRM assays. And that would then allow us to do drug screening tests on different perturbed systems. And I will be starting working on iPSC cells and different systems for that matter. And of course, the end goal is to get closer to validated assays for clinical proteomics. So to summarize this presentation, we have successfully implemented AFA-assisted digestion in our automated protein extraction workflow, and this really allowed us to increase the proteomic coverage, enhance the detection of membrane proteins in particular. We can now get deeper protein profile based on the detection of the lower abundant proteins, and we've significantly amplified the detection of mitochondrial proteins, which is of paramount importance in our other main projects. And this AFA-assisted digestion detergent-free workflow is also compatible on the 96-well plate assay, which is quite a feat itself. So if you're interested in learning more about the high-throughput cell-based assay system, I have a poster that I will be presenting on Thursday, as Samir mentioned. Poster number is 687. I've also attached the links and the QR codes to the Covaris channel to access some of the previous workshop presentations that I did in the past few years. So with that said, I wanted to thank Covaris and all the colleagues within Covaris, especially Samir and Eugenio, as well as Deb, for kindly allowing me to present once again. And from our lab, Max, Alejandro, Ali, as well as other members of the Van Eyck lab, and you guys for listening through this presentation. So with that said, I'm more than happy to take any questions at the end of the session. Thank you.